Today, we're going to be taking a glance at cells. We're going to be looking at their structure and function. We're even going to focus on not only their major attributes, but look at the major features of two main types of cells: prokaryotic cells and eukaryotic cells. So let's start first with microscopy. And what is it? Well, microscopy started in about the 1600s, and it was in order to view things that can't be seen with the naked eye. For example, cells have to be studied with microscopes. Cytology, the study of cells, was in its infancy in the 1600s. As early as the 1670s or the late 17th century, you had Anton van Leeuwenhoek who built his microscope, which was mostly a glorified magnifying glass, and looked a little bit like this. You could put a specimen on the point near the lens, you had screws and kind of knobs to help move about that specimen, but it was still very basic and rudimentary. Then Robert Hooke developed a primitive light microscope, and he observed cork. And cork really just looked like little boxes, and those boxes had a square shape. Robert Hooke noticed that those compartments, they resembled the ones monks would live in. And so those were called cells. So he called his cork boxes cells. So now that they had their moniker, that primitive inception was able to progress. In over 300 years, we have made great leaps and bounds in developing important tools and adding to the biological sciences, mainly by making microscopes better. While the first microscopes were invented in the 1600s, we've made leaps and bounds since then. Modern microscopes, of course, are more advanced. Light microscopes now use lenses made of glass or quartz. They bend, they focus light, they have better resolving power, so you can see the many levels of cells or tissues or specimens. We can even see structures inside living cells. Those can be visualized. Here are two basic microscopes you might see in a college lab. You have the light microscope on the left and the stereo microscope or dissecting scope on the right. The light microscope on the left, it has the best magnification out of the two of these microscopes. Dissecting microscopes have a much lower magnification, but they're really helpful to look at larger objects like tissues or just general objects that you might be looking at during a dissection. Here's a blown up image of a paramecium, and this is an image from a modern light micrograph. Keep in mind it has been blown up, it's much bigger than it would appear under a regular light microscope nowadays, but we can still see quite a bit of its features. It has hair-like structures on the outside that help it move. It has a contractile vacuole and fluid spaces and a nucleus. That's much more than we could see with those primitive microscopes we looked at first. But electron microscopes provide the best or the highest magnification and resolution than light microscopes. Electron microscopes actually use beams of electrons focused by magnetic fields to help create an image of different tissues or atoms or specimens. There's two different types of electron microscopes: transmission electron microscopes, or sometimes that's shortened to TEM, and they actually pass electrons through a thin specimen. So they let you see more of what's inside of a cell or specimen. You also have scanning electron microscopes, or shortened to SEM. They bounce electrons off specimens that are dry and hard, and that lets you see more of the topography or surface of your specimen. So while we have a light microscope to display living cells, it's really the scanning and transmission electron microscopes that reveal all the details, the ultrastructure of cells. We can really increase or utilize high magnification with electron microscopes. Resolution is more of a measure of clarity of an image, and we definitely have better clarity with electron microscopes as opposed to light microscopes. Electron microscopes also provide not only higher magnification, not only higher resolution, but just much more detail, that ultrastructure that we don't get from light microscopes. So here's an example of, again, that regular light microscope you might see in a lab on the left, and a picture of an electron microscope on the right. Again, there is a TEM and SEM, or SEM and TEM, microscope that we consider electron microscopes. The scanning electron microscope will give you a better idea of the surface structure of a specimen, and the TEM, or transmission electron microscope, would actually illuminate more of what's inside a specimen. Here's a better picture of the electron microscopy setup. That electron microscope is going to be hooked to a computer where those images populate. Those images are also going to be in black and white, So any colorized image from an electron microscope is just that. It's artificially colored. So here we can compare images from an SEM, scanning, and TEM, transmission electron microscope. Take a second and look at these two images. Both are showing up close and personal images of a pollen grain. One's from a scanning and one's from a transmission electron microscope. Which one do you think is from the scanning microscope? The left or the right? If you're thinking the left, you'd be correct. Scanning gives us an idea more about the surface. So we can see a lot of the details for the surface of these pollen grains in the image on the left. But a closer view of a single pollen grain allows us to see more of what's inside, and that's shown on the image on the right. And that's from a transmission electron microscope. Here's two other images, one from a transmission and one from a scanning electron microscope. Again, we can see inside using transmission, outside surface using scanning. Next, let's look at the cell theory. The cell theory states that all cells come from previously existing cells. Also states cells are the basic units of life. Once you have those basics down, we can look at the three components or principles of the cell theory. First, every organism is made up of one or more cells. So some organisms are unicellular, others are multicellular. We as humans are multicellular. And the smallest organisms are single cells, and cells are the functional units of multicellular organisms. That really plays off of principle one. So we can have more than one cell, and if there is more than one cell, those cells work together. And then last but not least, all cells come from pre-existing cells through processes of reproduction, whether that be mitosis or meiosis or other types of asexual or sexual reproduction. All cells arise from cells that were already here. There's also two basic types of cells, so that's what we're going to get into the next video. We're going to be looking at the basic types of cells, which are eukaryotic cells and prokaryotic cells.